Female palpation. Just as a disclaimer, all of the animal handling should be done in stocks and in a squeeze for safety, both animal and human. So, to begin, glove up the non-dominant hand with a lubricated palpation sleeve, grab the tail of the other and pull it backwards to help stabilize you. Then insert the gloved hand smoothly and quickly into the rectum. You will feel muscle contractions, but just push past that until you feel the pelvic bone kind of below your hand. Then you should also be able to feel for the cervix, which will be a cylindrical shaped structure, and to the left and right of that, a little farther out, you should be able to feel the ovaries, which should feel like little sacks with some marbles in them, depending on their stage of development. Another structure to feel for is the artery that will be running dorsally to the rectum, and you can bring your hand up to the top and feel for the pulsating blood.
Now we will be discussing the oocyte collection procedure. In this picture, you will see all the materials you need to successfully isolate an oocyte. To collect these oocytes, we aspirated multiple follicles using a needle and syringe from a bovine ovary collected from a slaughtered cow. The aspirate fluid for each aspiration was placed in a drop of saline solution, and the oocytes were located using a microscope. We were able to locate one oocyte from our aspirations. Once an oocyte is located, it is isolated by transferring it to a new drop of saline. Once it is washed to remove any extraneous debris, it will be ready for any further IVF procedures.
Male palpation. Similar to female, glove up with a lubricated palpation sleeve on non-dominant hand, grab tail with the right hand, and pull backwards to stabilize. Then insert hand smoothly and steadily into the rectum. You do not have to go as far in as with the cow. Feeling for the pelvic bone again is your landmark, and from there you can try to feel the seminal vesicles, which depending on age and if they are inflamed or not, can feel like squishy grapes all the way to little rocks, which would not be good, indicating infection or inflammation and a blockage. Measuring the scrotal circumference is also important because scrotal circumference will contribute to the inner volume of the tissue. More testicular tissue means more ability to make sperm. This is done by placing a measuring tape around the scrotum and making sure the testes do not ascend back up into the body with your hand.
Our next step is semen collection. Here we are collecting semen using electroejaculation. A lubricated probe is inserted into the bull's rectum so it sits adjacent to the prostate gland. This insertion is done right after the reproductive tract has been rectally palpated. Once fully inserted, the operator can leave the stock area and use the bull's tail to keep the probe in place. The electroejaculator is turned on, and the voltage is gradually increased until the bull protrudes the penis and commences to ejaculate seminal fluid. A person stands by to collect the semen using a sleeve that is hooked on a loop apparatus. The fluid that is initially released does not contain any semen, and so the semen collector waits a bit before catching the ejaculate in order to obtain a more concentrated sample. They then place the sleeve over the tip of the penis and follow the bull's movements in an attempt to catch as much semen as possible. Once the bull has finished ejaculating, the semen collector steps aside and the electroejaculator is removed.
Semen analysis. There are many things to check for in semen analysis, but the first one that you can check for right after collection is the color and if there is any debris in the collection, which can be used as those preliminary criteria. So the collection should not be thick, which it would indicate that there would be an abundance of white cells, which could mean a possible infection, and it should not be yellow in color, which could mean the presence of urine, which would mean that those sperm would already be dead, so it should be an opaque whitish color. And a sample can be drawn up either from the raw semen or it can be extended there. Then it can be placed onto a wet or dry mount on the slide to be viewed under a microscope, where it will be further evaluated for motility, morphology, and concentration.
No, so cleaning's for the vulva. Yeah.
We are artificially inseminating cows. Fiona and I just went, but we're going to coach now.
So lubed up, going in rectally.
We've loaded up the guns with semen, and first you're gonna insert your left hand into the rectum and find the cervix.
Can you hold mine too? Yes.
Delightful.
After locating the cervix, you will use clean paper towels to wipe off the exterior of the cow's vulva. Once the vulva is clean, you can begin to insert the straw into the vagina, beginning at a 45-degree angle and then moving along until the straw is parallel to the ground. Once the straw hits the hand that is holding the cervix, you can maneuver it into the cervix until the end is near the uterus. Then you can inject the semen into the uterus and withdraw the straw.
Placenta examination. First, observe the front and the back of your given reproduction tract, the ovaries, oviduct, uterine horn, and body of the uterus down to the cervix. Then start by cutting the cervix. Use full lengths of the scissors to make your cut, and try to avoid rupturing anything internally. Keep cutting up and over the middle of the pregnant horn, which will be the larger, more developed one. The other will seem shrunken in comparison. Once that is cut open, you should be able to see the fetus and the placenta around it, with its caruncles and cotyledons, which are the points of blood and hence nutrient transport between the fetus and dam. Gently try to peel back the outermost layer, which is the chorion. The next surrounding sac is the allantois, which is composed of all the fetal waste, and the innermost sac is the amnion, which provides most protection for the fetus. You can then examine the fetus as well.